Hello, everyone. In today's talk, I will share with you the GATK-for-Microbes project, which is an improved GATK method for variant discovery in microbial species. GATK stands for Genome Analysis Toolkit. It is an open source software package with over 240 tools and more than 75,000 users. It is used to discover variants after reads come off the sequencer. However, GATK was primarily designed for human data and was not envisioned for microbial genomes. Despite that, we noticed that the community was already adopting GATK for microbial variant analysis. Here is a timeline of the number of papers published on microbial research that use GATK. One example of this is SnapperDB, which uses GATK to generate a bacterial variant database used by the England Public Health Department for pathogen surveillance. Study of Enterobacter strains isolated from the International Space Station, outbreak analysis of Klebsiella, and various other highly important research. Seeing how the microbial community was already adopting GATK, we decided to develop improved and accurate GATK methods specifically intended for microbes. Let's talk about some of the challenges with optimizing GATK for microbial genomes. First, microbial species can have variable ploidy, and many have circular closed genomes, while human genomes are linear and diploid. Microbial genomes have very high genomic diversity. Additionally, microbial genomes are much smaller in size compared to human genomes, and therefore, sequencing and variant calling errors made with microbial genomes are significantly more important to deal with. Here are some of the high-level improvements we have made in GATK. During our investigation, we noticed that the microbial community was adopting GATK's germline variant discovery tool called haplotype caller. However, our research found GATK's somatic variant caller, MuTec2, does much better with microbial genomes. We developed a tool to optimize for the closed circular genome structures of microbes. Additionally, we have made some improvements to the GATK algorithm itself for better sensitivity and precision. So let's talk about the improvements we've made to the GATK's algorithm. The current GATK's local assembly-based variant caller uses Pileup data to identify regions of the genome that have evidence of variation. These are called active regions. GATK creates a de novo assembly of the reads overlapping the active regions, from which it derives a list of potential haplotypes and the most likely genotypes. This method has worked very well for human data, but we've noticed that the assembly method can fail in some regions that are complex or have a lot of variance. To remedy this, we developed a hybrid GATK method which combines local assembly and Pileup-based variant calling methods. In the hybrid method, we generate additional Pileup-based haplotypes. This hybrid method has drastically improved the sensitivity of GATK on microbial data. This is one example of where the assembly-based method fails, but the Pileup method rescues the variants. The first track in this IGB screenshot is the truth variance track, which we use for sensitivity evaluation. The second track is the GATK's assembly-based method, which does not identify any variants. And the reason is that when variant events are clustered like this, the assembly graph becomes very large and GATK is unable to process it. However, the Pileup-based method is able to identify and rescue such events, and you can see that in the third track. We tested our new pipeline on real fungal data and compared it to other variant callers such as Pylon, Freebase, and GATK's germline tool, haplotype caller. We found that our new GATK-for-Microbes methods does better than other callers in terms of sensitivity. But as you can see in the precision column, we still have some work to do. We are currently working on methods to improve our precision. And finally, we are using cloud-based methods to enable global collaboration of data and results. We have made the GATK-for-Microbes pipeline available in Terra. Terra is our cloud-native platform developed by the Broad Institute in collaboration with Microsoft and Verily. Last but not the least, we have a lot of people to thank for the success of this project. And please feel free to contact us if you have any questions. Thank you.